Hello everyone, my name is Dr. Graham Cottrell and welcome to the final video on the molecules of life relating to proteins. So in this next video we're going to explain how we know that the structure and function of a protein is encoded within its amino acid sequence. And we're also going to discuss how human diseases are caused by mutations in proteins. So mutations in a DNA sequence, we all know that now that DNA encodes for the protein, can lead to changes in the primary structure of a protein. So that even a single base change from an adenine to a thymine can lead to a different amino acid being incorporated into the protein. And this change in the primary sequence can lead to a loss of function, so the protein or enzyme may not work properly. It could cause the protein to misfold, or it can, in some cases, actually cause what we call a gain of function. So this can lead to a protein having a function that it doesn't normally have. One of these common diseases is sickle cell disease. So this is an inherited condition. So it's passed on from to your children that affects the red blood cells of the body. And what this does is it's a mutation in the beta globin gene. And this is a single base change. Here we can see from an adenine to a thymine that causes the replacement of glutamic acid with a valine. Now a normal healthy red blood cell will have this sort of almost donut shape with not the hole in it. It's got a little dimple in here. But that single amino acid change from glutamate to valine will cause a complete different folding of the beta globin. This means that the red blood cells, which normally flow through your blood vessels unrestricted, tend to sort of clog and struggle to get through, so they don't go through very smoothly. Another disease that's caused by gene mutations in genes is cystic fibrosis. So the cystic fibrosis gene is a chloride channel. So normally in a healthy lung cell, this protein, the CFTR, cystic fibrosis channel, allows chloride to pass through freely. But then we do have a single mutation, and here the example is a glycine at position 100 is 551 is replaced with D, which is a aspartic acid. So we're going from glycine, which is a neutrally charged amino acid, because this on the R group just has a hydrogen atom, to aspartic acid, which will have a carboxyl. So we're getting a negatively charged amino acid there. And what that does is it causes the protein to fold incorrectly, or the charges are incorrectly. You can imagine that the chloride ions, which are negatively charged, are trying to get through, but you've introduced a negative charge into the protein, which is in ported position, and so this will repel the chloride. It won't allow it through, and this causes the buildup of thick and sticky mucus. We also have mutations where an amino acid can actually be deleted. So this is a common, what they call delta F08, and what this means is that the protein is made normally, but it doesn't get to the position in the cell where it needs to. So the mutant protein needs what we call a corrected drug, and what this does is it allows the channel then to go to the cell surface where it can actually function properly. So two different types of mutation leading to the same disease, we have a simple amino acid change from glycine to aspartic acid, or we call the delta F508 mutation. Lactose intolerance is also caused by mutations in a particular gene as well. So this gives the person an impaired ability to digest lactose, a sugar which is found in milk. Later in the course we will be doing a practical based around lactose. So lactose intolerance in infants is caused by mutations in the gene, whereas lactose intolerance in adults is generally due to basically a decreased expression. So as you get older, some people will lose the ability to make the protein, and this is not mutations in the protein itself, but these are mutations in a regulatory element. So these are upstream of the open reading frame, and this will gradually lead to less and less of the enzyme lactase being produced. So supplements can be taken to combat this, and again we will cover this in more detail in a practical in spring term. Alcohol intolerance. So some people are not able to drink alcohol because they have a deficiency in an enzyme called aldehyde dehydrogenase 2. And this enzyme is responsible for the breakdown of a toxic compound, but as your body digests the alcohol, called acetaldehyde. And this is quite a common deficiency, especially in people of East Asian descent. And there is a single point mutation in the enzyme, and this reduces the activity, so that the people with this mutation are unable to convert the acetaldehyde to acetic acid. This is a toxic chemical, and if it builds up, then it can lead to the patient being very ill. Alzheimer's disease is also a disease associated with mutations in not only amino acid sequence of the protein, but also in folding. And this is caused by the accumulation of a peptide called A-beta. So in the body there is a large protein which we call the amyloid precursor protein, or APP for short. And this is present on the cell surface of the cells in the brain. Enzymes cleave this, and this short form here, coloured in sort of orangey brown, is released outside the cell, and it's build-up of this protein that they all combine together, and they form insoluble fibrils, and they are composed mainly of beta sheets. And it's the build-up of this which causes neuronal cell death. So when it's part of the APP, everything is fine, but there are enzymes that cleave the N and C terminus of the short peptide. This then builds up, forms plaques in the brain, and leads to neuronal cell death. Prion disease is another disease which is caused by misfolding proteins. So this is quite an unusual disease. Prions are protein molecules that basically cause disease. So you get interaction of an infectious prion molecule. So this is a protein that for some reason hasn't folded correctly. And when this interacts with a normal prion protein, it causes that protein to also misfold. So we have the non-infectious one here, and we have the infectious one at the top. And when they interact, it causes both proteins to be completely misfolded. Then there is a chain reaction, and you get more and more of the misfolded proteins, which can lead to the buildup of which causes prion disease. Proteins are made of regions which we term domains, and these can quite often have their own particular fold or particular function. So one domain of a protein may have, say, a kinase domain, which is responsible for phosphorylation of proteins. And domains can be isolated away from the protein, and they will fold independently on their own into an active conformation. Most proteins are made up of a number of identifiable domains, so these are discrete, and we could draw these out. And it's these different combinations of domains that give many proteins their unique properties. So each domain of a protein will have a specific function. An example here of proteins containing their domains are the immunoglobulins. So they have what are called constant and variable domains. And you can see here these constant domains in blue. And then we have the variable domains here. Immunoglobulins are an example of a protein which is made up of various domains. We can see these constant domains here, CH3, CH2, CH1 and the Cl, so these are constant. So what this means is that all antibodies or all immunoglobulins from a particular person will have the same amino acid sequence. Whereas these variable domains, these are the portions of the antibody that will bind specifically to an antigen. These are variable, so these will change depending on what the body wants the antibody to bind to. So each of the domains of a immunoglobulin will have specific functions, and here highlighted we have the antigen binding domains. These are domains at the bottom here are for binding to what we call Fc receptors, and then there are other domains which are responsible for placental transfer. So this is when the mother will pass antibodies on to the growing fetus. And then we also have a specific domain which will recognize a complement binding site. Many proteins, after they are made, undergo what we call post-translational modifications. So this is where different molecules are added to the proteins after they've been made. A very common one, and you should remember this, that this occurs in the Golgi apparatus, is that proteins are glycosylated, so sugar groups are added to the proteins. And this can help with the conformation, it can alter the subcellular distribution, but they're also important for stability and the activity of the protein. Many proteins will undergo a phosphorylation event, so this is where phosphate groups are usually added to serine or threonine or tyrosine residues. So this is quite often an event that happens during a signaling cascade. Many proteins are tagged with a protein called ubiquitin, and this process is termed ubiquitination. Ubiquitin is a 76 amino acid small protein that's added to lysine residues in a protein, and what this does is it targets it for degradation either by the lysosome or by a intracellular cytosolic protein complex called the proteasome. Some proteins are also nitrosylated, and this serves to regulate the gene expression. It can stabilize the proteins so they are longer lived, but it can also mean that they can provide nitric oxide, which is an important signaling molecule. Ugh. Other types of modification include methylation, which is important in the epigenetic regulation of gene transcription. Cetylation, again, plays an important role in gene transcription. Lipidation, so this is important for targeting protein molecules to the cell surface. So if a protein has a small lipid group on it, this lipid can then insert into the plasma membrane, basically tethering the protein to the cell surface, and this serves to localize its activity in a particular place. Many proteins are also proteolytically processed, so this can be to activate them. So an example here is trypsin. So trypsin is an enzyme which your body uses to digest your food, but this is produced in the pancreas as trypsinogen, so this is an inactive form. But once trypsin reaches the place where it needs to be active in your small intestine, then it's actually proteolytically cleaved by another protease, which then releases the trypsin, which is then the active form of the enzyme. So here's a multiple choice question that could be present in an examination. So which of the following post-translational modification involves the hydrolysis of a covalent bond present in a protein? If you want to have a go, maybe you want to pause the video now. Of course, the answer here is proteolysis. Proteolysis is the breakdown of a peptide bond, and this is the covalent bond present in a protein. Glycosylation is addition of carbohydrate groups. Methylation, addition of a methyl group. Lipidation is addition of a fat. And ubiquitination targets proteins for degradation. An additional reading again can be found in chapter 3 of Albert's Molecular Biology of the Cell and in chapter 2 of Stryer in Biochemistry. Thank you all for listening.